All right. Hi, everyone. So today we have Caleb Heaslip here to give a presentation. Caleb is a third-year chemistry major, and he took organic chemistry with me last year. So yeah, I can see some of your friends are here. And yeah, and Caleb did a summer internship at University of Michigan, and he's going to talk about what he did there. And for those of you who are in the second year, this is something that you might be doing in a year. So Caleb, the floor is yours.
Everyone hear me pretty well? It's not too loud. Okay. So yeah, can people on Zoom hear me pretty well? We get like a chat. Can people on Zoom hear me pretty well? Or I just want to make sure. Okay. Okay, awesome. So yeah, for those of you who don't really know me, my name is Caleb Heaslip. As Dr. Shia just said, I'm a third-year chemistry major, and I was fortunate enough to. God, yeah, I was fortunate enough to spend the summer at the University of Michigan's medical school. I participated in the cancer biology research program. It was a great experience, and I'll share some of the research I did in the data, and then afterwards I can maybe expound more upon my experience there, and what I learned as a budding scientist, and as a future communicator of science, hopefully. I consider myself pretty fortunate because I've found a real I guess passion for being a physician scientist and advancing medicine through research, but also through clinical practice. And what I researched this summer was diffuse intrinsic pontine glioma, and more importantly, targeting the metabolic vulnerabilities in that disease. So the lab I worked for, Dr. Dan Wall, he's a radiation oncologist at the University of Michigan, and the overarching kind of goal of the lab is to understand how altered metabolism contributes to resistance to radiation therapy in high-grade brain tumors. So what is DIPG? So DIPG, or diffuse intrinsic pontine glioma, is a very, very rare, highly aggressive malignant brain tumor that occurs primarily in children. It occurs in the brainstem, being the most common pediatric brainstem tumor. And I'll get more into that. There is a significance to the actual anatomical location of where these tumors arise, especially as you go from pediatric to adult cases. But it's an extremely sensitive area, and the outcomes are extremely, extremely poor. It's a dismal diagnosis. It's essentially a death sentence. You can see the numbers there, with the median survival only being about 11 months and a less than 1% 5-year survival rate. So there's a real urgent need for therapeutic improvements in this disease. And this is the leading cause of brain tumor related death in children. So kind of contributing to these really poor outcomes we see are a few key obstacles to treating DIPG. So first is that radiation therapy is the current standard of care. It's actually the only standard of care that's demonstrated any efficacy in patients. So it's actually quite interesting. For the first 4, 5, 6 months of radiation treatment, the tumor size shrinks, clinical symptoms decrease and start to ameliorate. But at that kind of 5, 6 month inflection point, radiation just stops being effective at all. And in fact, some research has shown in vitro actually that radiation can actually help cells to proliferate and grow even faster. So it's a really difficult obstacle, and I'll get later into the presentation, actually how cells acquire that resistance to radiation, even hijack it. There's a lot of kind of metabolic implications in that. Number 2 is that surgical resection is not an option for 2 reasons. This is a major limiting factor as well, is that number 1 is that DIPG occurs in the pons. If you think about where the pons is in the brainstem, many critical basic functions are routed through that area. So if you think about, your respiration rate, the coordination of your circadian rhythm and your sleep-wake cycle. So that's an area that upon perturbation of healthy tissue there, you can have severe, toxicities and symptoms associated with that. And number two is in the actual name of the tumor, its diffuse nature. So this tumor doesn't grow as like a robust mass you can kind of scoop out. It really weaves its way through healthy tissue. In the image I liked, I used this summer to kind of visualize that is when you have a ball of Play-Doh and you mix the colors together, really well. I'm sure you've done that as a kid. And, looking at that picture, it's very hard to find like a demarcation line between the different colors. If you were to try to separate those colors, it'd be very difficult to do so without doing considerable damage to the overall mass itself. So that poses a substantial barrier. And then last but not least is that chemotherapeutic strategies have been essentially ineffective at all. There's some efficacy that's been demonstrated in mice and in vitro, but when it comes to clinical trials in patients, it just hasn't been enough. It's essentially like you're giving them a placebo, honestly. So it's a really difficult problem. And before I get into this further, really just shout out to the families of these patients, because they've really helped to propel this field forward by being vocal about it. Because unfortunately, chemotherapeutic development for pediatric cancers tends to be behind, for development for adult cancers. That's due to many reasons, economical, in terms of case volume. These are rare cancers. Only 200 to 300 children a year get DIPG, so it's hard to run large-scale clinical trials. So there's not a ton of data available, but really the families of patients have made the research I did this summer possible. And contributing to foundations and funding this research is really important, and that's largely spearheaded by them. So, okay, and then also a special shout out to two very incredible researchers who about 10 years ago really ignited a newfound interest in understanding this disease. These are doctors Nana Jabato from McGill University and Susie Baker from St. Jude's Children's Hospital. They both independently discovered the key driver mutation for this disease, which I'll get into right after this. But without their research, what I did this summer would not be possible, because my research was completely in the context of this mutation. And they're really leading figures in the field. And like I said, the progress they've made has really kind of helped to energize new research and understanding, but there's a long way to go. I remember listening to a lecture to Dr. Jabato, and she described it as this understanding was a transition from the Ice Age to the Stone Age. And the fact that, tackling this disease, we're essentially still trying to take down woolly mammoths with stones from, slingshots. There's a lot more to be done. So. And yeah, these are just those two kind of seminal papers pointing out that histone mutation that you may have read in the abstract that I'll get into shortly. But these two papers were essentially my Bible over the summer that I referred to, the one on the left being from Dr. Baker's lab and the one on the right being from Dr. Jabato's lab. Again, this ignited, this new understanding into the molecular profile of this disease. Okay, and this is really interesting. I'm not going to get too into it because it's still pretty complex, but DIPG in high-grade malignant brain cancers, for that matter, are really a manifestation of abnormal development. And to visualize this, again, this is really neat, is that the far majority of pediatric brain cancers, as you see from the number there, 78%, occur in the midline area. And as the age of prognosis for malignant brain cancers gets older, they literally kind of traverse along a longitudinal axis to the brain, to the from the midbrain to the cortex. It's really interesting because that suggests there may be a developmental basis to this disease. If you think of children, their brain is less developed. They have less kind of neuronal structures established in their cortex. Those tumors are all happening in the midline and the brainstem. So your pons, for example, your thalamus, DIPG is labeled there, and the pons with that K27M mutation, which I'll get into. But adult cancers, the far majority of adult malignant brain cancers occur in the cortex, which is that kind of cotton looking like thing that encapsulates the brain. That's mostly where I'm sure you've heard of like glioblastoma occurs. So it's a really kind of an interesting thing to look at. And here's a quote from Dr. Jabato. In that pediatric and young adult astrocytomas are developmental defects and a story of 2 lysines on H3 variants, which again I will get into very shortly. But that's an interesting picture. That's something that I didn't even know about until this summer, and it's really unique to look at. Okay, so a little review for molecular biology, hoping to make Dr. Ellis proud. The key to understanding this disease is to first, understand that epigenetic modifications are key for regulating gene expression. DNA does not exist in isolation in the nucleus. They're actually associated with proteins called histones, which facilitate the extent of DNA packaging. And it actually provides a physical basis for gene expression. So, if you think of DNA as, yarn, when DNA exists as like a loose thread of yarn nucleus, those histone associations, they're not tightly associated to the DNA. That DNA is freely available to be bound to by, transcription factors and all that, good machinery to express genes. But when genes are silenced, and these are promoted via various modifications to the histones, gene silencing is like taking that ball of yarn and like wrapping it up. So the DNA physically isn't available to be bound to by that transcription machinery, and thus genes are silenced. But these processes are highly complex and dynamic. But an important thing to remember, and this is offers a potential kind of therapeutic direction for drug development, is that these modifications that facilitate this packaging, they are reversible. Okay. There's your molecular biology review. And then time to get into what I've been referring to. So 80% of DIPG tumors are characterized by this single H3K27M mutation. And what this means is that on the lysine 27 residue of histone 3, there's 4 different types of histones, each has a few variants. Like I said, those are the proteins that associate to DNA. In this histone 3, that lysine 27 residue on that tail of the protein, we have a change from a lysine amino acid to methionine amino acid. That is it. And that single mutation is the primary oncogenic driver of this disease. And it leads to, this horrible tumor, which I find fascinating, is that a single amino acid can make such a difference. But, this site is, a very common site as well for histone and epigenetic modifications and regulations. And specifically, as I just mentioned, this histone 3 lysine 27 site is a very common epigenetic marker. Methylation, for example, promotes gene silencing and the condensation of DNA to form heterochromatin. Acetylation typically exposes DNA for the formation of euchromatin, and hence you have active gene expression. But specifically, what we're looking at is the trimethylation, so adding 3 methyl groups to this lysine residue leads to heterochromatin formation. DNA becomes tightly associated. Those gene promoters are inactive and they're unavailable to that transcription machinery. In this single mutation, as I'll get into the mechanism after more specifically, this promotes this tumorigenic character. They term post this mutation, the term for this would be an oncohistone because it contributes to the formation of cancer. So how it does this specifically is that this mutation, it blocks the catalytic action of this enzymatic complex called PRC2. It stands for polycomb repressive complex 2. And it blocks this complex's ability to methylate a region of that genome at that site. So, this complex is, under normal conditions is adding those, di and trimethyl groups, but this mutation just puts a traffic cone there and it's halted in its ability to catalyze that methylation downstream. So essentially what you have in this disease is a region of the genome that under normal conditions without this mutation, it's all trimethylated and the genes are inactive. There's no active gene expression going on. But with this mutation, we have DNA being inappropriately opened up and exposed, and hence you have gene expression that's occurring, very likely connected to developmental genes, as I suggested in, a few slides ago. And this is the primary contributor to the development of this tumor. And now into more of what the umbrella that my project is under. As I mentioned, the lab I'm in is interested in looking at how altered metabolism contributes to resistance to radiation therapy. And this is actually some data from one of our collaborators at the University of Michigan. I'm not going to get too into that. Essentially, it's just showing that this mutation induces a unique metabolic profile. As you see, looking at the wild type on the left column and the cells with the mutation on the right column, you see that stark difference there. When you see that checkerboard pattern with metabolic data, you have some heterogeneity between that. And this unique metabolism, it's important because metabolism, you think there's many metabolites involved, there's pathways, so these are targetable. And that's what I was looking at in my project. And this is more kind of metabolic data that the graduate student, Eric Peterson, that I worked with this summer, worked on right before I started. Again, not going to get too much into this, but essentially what he read from this data is that some of the most pronounced changes in metabolite levels from wild type DIPG tumors to the tumors with that H3K27M histone mutation, a lot of these metabolites we're seeing are involved in purine pathways. They're, purine metabolites, purine synthesis, which I'll get more into. But those are the key metabolite differences that, kind of sparked the idea of his project, which I helped him on this summer. And before I get into that, I know I mentioned, radiation therapy earlier. So radiation therapy actually kills cancerous cells by damaging DNA, and that's either directly or indirectly. So the direct mechanism is that it actually just breaks DNA. That ionizing radiation, it induces single or double-stranded breaks, with double-stranded breaks being more potent in terms of toxicity and harm to the cell. And also indirectly by inducing the formation of free radicals, which also leads to considerable DNA damage as well. And an important note is that cells acquire resistance to this radiation through many mechanisms, which again for researchers and for people interested in drug development makes it extremely difficult. Because like I said, that radiation, it works awesome for the first 4, 5, 6 months in this disease, but then it just stops. It's pretty incredible how cells are able to do that, but that's what fuels this new research. And specifically, some of these mechanisms to radio resistance involve metabolism. So radiation therapy can induce many alterations in the metabolite molecules in the metabolic pathways themselves. And many studies as well have suggested a link between tumor metabolism and resistance to radiation therapy. And this is actually a study that I'll get into later in my presentation that the Wall Lab actually published in the context of glioblastoma, which is an adult high-grade brain cancer. But this is still a very active area of research. There's still a lot to know. Again, these metabolic pathways and how they actually, connect to this idea of radiation resistance is still complex and largely unknown. Okay, so I mentioned the most pronounced metabolic differences in purine levels. And so just kind of quickly go over, the importance of purines. If you picture a cancer cell, and it gets damaged by DNA, how does it stay alive? Well, it needs to repair that DNA, and it needs the components and basic building blocks to further proliferate. And those are purines. If you think of your adenine and your guanine, again, from your molecular biology, those are the basic building blocks of DNA. You can't, build a house without, wooden planks and, your steel beams or whatever. That's what purines are. But purines are also really important energy carrier molecules. If you think of ATP, GTP, cyclic AMP, that are heavily involved in all these metabolic pathways in giving the cell, that sustenance and energy to repair that DNA and carry out those processes. And this too, specifically in the context of the application of radiation therapy, is still kind of unknown, the other role of purines, because obviously, like I said, they're the building blocks of DNA. But one of the members of the wall lab that I was in this summer is actually actively studying this, how specifically purines are involved in DNA damage response pathways, specifically as signaling molecules like GTP and ATP. Okay, and then how are purines actually synthesized? Well, there's two primary mechanisms. That's a typo there. Just don't look at it. De novo and salvage pathways. So de novo is making purines from scratch. And salvage, as it sounds, is recycling purines back through that pathway to make your GMP and AMP. And that's a good little graphic that I got of de novo purine synthesis on the top. Then on the bottom, you can see adenine, hypoxanthine, guanine. Those are purines being funneled back through. And there you get your GMP and AMP, which upon further phosphorylation, you get GTP, ATP, and then your nucleosides as well through being funneled into other pathways. Okay, so now this is the details of the project I'm working on. So what we found with that metabolic data specifically is that in the DIPG cells with this H3K27N tumor, which as I said constitutes around 80% of the tumors in this disease, we found an abundance of hypoxanthine and guanine, which are these two metabolites right here, and a depletion of guanosine. So what that told us is that, so these metabolites are being accumulated, so they're not being used up. So maybe there's something wrong with that HGPRT enzyme that's catalyzing their conversion and funneling them further into this pathway to synthesize the purines. So yep, there's that enzyme right there. So to kind of confirm this hypothesis, I did a Western blot to quantify this expression of this HGPRT enzyme, the gene being HPRT1. And essentially, I quantified that data, accounting for differences in backgrounds and kind of the protein bands. For those of you who've done Western blots, you're probably familiar with this, essentially into an expression ratio. And what this tells us is that, the hypothesis was correct in that the DIPG13, so those are the two different cell lines we use. The DIPG cells with that H3K27 mutation express a lower level of that enzyme compared to wild type, which accounts for that abundance we saw in those metabolites. And something also worthy of note is the heterogeneity between different cell lines. Again, not to get too much off on a tangent, but that's a really difficult thing in cancer as well, because not only are different cancers so different from each other and there's not much overlap, makes targeting them very difficult, but even different cell lines and cells between patients of the same cancer are considerably different at times. That's something we're studying in the lab and working at now at Dana-Farber, trying to target cells and trying to find common overlap between them, because you would really be surprised, how by such orders of magnitude how they differ in the same cancer, which again makes this a very, very difficult problem to address. Okay, so we've established that. So there seems to be that deficiency in that purine salvage pathway. So that enzyme that's taking those metabolites, funneling through the pathway, there's some deficiency in that. So we kind of thought, if that purine salvage pathway can't supply those purines, then maybe by targeting de novo synthesis, which is the other modality of synthesizing purines, maybe we can, completely, cripple the purine supply in cancer cells so they can't repair that DNA damage from the radiation and, subsequently, become radio resistant. And so we applied a drug, MPA or mycophenolic acid, which is an IMPDH inhibitor. IMPDH is a key enzyme involved in that de novo pathway from the conversion of IMP to GMP, I'm pretty sure. But yeah, so again, we have those two pathways of synthesizing purines. They have some natural deficiency in one, so we're targeting the other to hopefully shut it off and make them susceptible to radiation. And so this is the assay that I conducted. It was a neurosphere assay. So essentially, day 0, out a bunch of cells, applied the drug to those cells, waited 24 hours. Then day 1, we irradiate, let it incubate for 24 hours, plate into a 96 well plate, and then incubate for a week, and then quantify the cell counts using cell titer glow assay. For those of you who aren't familiar, cell titer glow, it's an assay to essentially quantify cell count. So the cell titer glow reagent, it reacts with ATP, produces a luminescent signal that you essentially read through machines. The idea is that, the more signal you have, the cell count, there's a higher cell count because cells that are dead are not going to be metabolically active, hence they're not producing ATP. And I should qualify that by saying the only reason you can make that conclusion is by pipetting the same number of cells into each well. So yeah. And then that's a little more insight into the setup I did. So we have 3 different drug concentrations, a 0 micromolar control, then 1 and 10. And then within each of those groups, we have 4 different radiation doses, 0 gray, 2, 4, and 6 gray. So 12 groups in total that gave us a decent amount of data to work with and kind of form those conclusions. So this is the data I collected, and we found that MPA actually it increases sensitivity to radiation therapy. If you look at that graph on the top left, that's the normalized data as a percent of the no radiation therapy control. You see at that 1 micromolar MPA, it really plummets down to 50 after that first hit with radiation. Hence, the cells are becoming more susceptible to the radiation. I will point out that if you look at the graph in the top right, so that's kind of plotting the concentration of the drug as a function or to the toxicity. And you see we were having problems at that 10 micromolar concentration. It was extremely cytotoxic to the cells. They were essentially just plummeting even without radiation applied. So I didn't include that data there. But essentially what it would have looked like is that on this graph on the bottom right, that initial luminescence read for that 10 micromolar MPA was like half of the others. So it was hard to normalize the data. And then on the bottom left, that enhancement ratio is essentially just quantifying how the cells respond to the radiation. So with applying the drug, this MPA, with radiation, it's substantially, the decrease is more substantial compared to without any drug. And again, I should have prefaced this by saying all the research in this lab was in the context of combining chemotherapy with radiation therapy. Because as I said, radiation therapy is the current and only standard of care that demonstrates some efficacy. So any trials, testing new chemotherapeutic regimens, the ideal, most effective is in combination. So, radiation therapy plus chemo. And so this is just the same data, but with a different cell line I was working with. And again, it kind of confirms the same thing in that this MPA increases the sensitivity to radiation therapy. You can see again, after that first hit with radiation, it really knocks it down more than without any drug. Looking at that enhancement ratio again, it's showing that these DIPG cells with the drug applied are are much more affected by radiation to a greater extent. And again, at the top right, we still see that same toxicity with the 10 micromolar. Towards the end, I didn't have time during my internship to really kind of account for that in the sense that, so as I said, we applied the drug for 24 hours and then we would irradiate. 24 hours later, we would plate to the 96 wells. That procedure, we washed off the drug. So the drug was on for 48 hours. And towards the end, when I was meeting with my mentor, we're kind of like, okay, maybe we're leaving the drug on for too long. So then towards the end, I tried experiments where, for example, I would apply the drug at 9 in the morning. 6 hours later, I would irradiate. Then at like 9 the next day, I would wash and plate to the 96 well. So the drug in that sense was only on for 24 hours. I only did that experiment like once, got kind of wacky data. And again, that, that happened for these experiments a lot. And I could kind of talk about it after, you get some wacky data and, you just have to stick with it. But, I think if I repeated that more times and, had some more time, I could have, gotten some good data out of that. Okay. And so what's pretty cool about this project is that, it's kind of confirming a hypothesis that the Wall Lab actually published. And they found a very, very similar thing in that purine metabolism seems to regulate DNA repair and radioresistance in glioblastoma, which is an adult high-grade malignant brain tumor that occurs in the cortex. And they found that, these metabolites correlate significantly with radiation resistance. And the important thing is that this was found across genomically distinct cell lines of GBM. So as I mentioned earlier, there's a lot of heterogeneity between cell lines of the same tumor. And what's important about targeting this purine metabolism is that no matter what your differences in phenotype are, to repair DNA and to proliferate, you need purines. So hopefully targeting purine metabolism, as this project shows, as well as this paper that was published in 2020 by the Wall Lab, is that this can offer a new method of treating these brain tumors that again have dismal outcomes and really need therapeutic improvements. So yeah, DNA repair was inhibited by specifically targeting GTP synthesis. You can see in that figure in the top left, essentially the same thing. They applied the same drug to that IMPDH enzyme, which downstream of that produces GMP. Upon further phosphorylation, you get GTP. So again, it makes sense, inhibiting that synthesis of GTP, which can then lead to guanosine nucleotides. Those cells were hampered in their ability to repair that DNA after radiation therapy. Okay, so kind of the key takeaways from this is that this H3K27M mutation promotes altered purine metabolism compared to the wild type tumors. Again, this mutation is found in 80% of these tumors. It's pretty fascinating how such a large portion of this disease is really just ignited by that single amino acid mutation. It makes it very interesting to research, but little disheartening the fact that only one mutation and the decades of research have really struggled to address that and improve outcomes for patients. This project showed that in vitro targeting of de novo purine synthesis using MPA increases radiosensitivity to H3K27M mutant DIPG tumors. And again, as I just showed, prior research, out of the Wall Lab especially, has established that inhibiting purine synthesis may be a potential strategy to treat these high-grade glioma tumors. And I guess the final takeaway is that therapeutic targeting of purine metabolism in combination with radiation therapy is a promising strategy for improving much needed outcomes in this disease. So just for a few acknowledgements, specifically Dr. Dan Wall, who is my principal investigator. As I said, he's a radiation oncologist at the University of Michigan. He was really generous this summer, with making me feel part of the team and offering all his insights. And I got to shadow him a few times and really, immerse myself in that lifestyle of what being a physician scientist looks like. And it really only strengthened my desire to pursue this career. And additionally, Eric Peterson, who's a graduate student in the cancer biology program, and he's who I worked with very closely this summer. Again, this is my first experience doing like wet wet lab research, doing bench work. And he was super patient with me. I'm not going to mention the Western blot that, I did the membrane transfer in the wrong direction, even though I just mentioned that. But yeah, all the wall lab members right there, CARSIP program, the people kind of helped my onboarding. And what this taught me, I couldn't say, enough, positive things about University of Michigan. It was, everybody there was super humble. Every other week I was having a lab meeting with, a real kind of pioneer and leader in a field, people who are Howard Hughes medical investigators. And, there's really just no ego attached onto it at all. So it was really, really great experience. And here's a few action shots, me representing MCPHS, in that left photo, that was one of our bi-weekly luncheons. So every 2 weeks, a graduate student would present their research and we all have lunch together. I'm actually wearing this same sweater right now. And then, my PI, Dr. Wall, he was kind enough to give me a little shout out on Twitter. I feel bad, I don't have a Twitter account, so he couldn't tag me to it, but maybe that was a sign that I should get more connected on social media, I guess. But yeah, that was me delivering that presentation. I only had 12 to 15 minutes to do it, so I was kind of racing through it. But yeah, I guess, I can go over some questions, maybe discuss a little more. Expound upon my experiences, a bit more in depth. And yeah.
I'd like to go back to your MPA readings, because you mentioned you did radiation and then measured, and you had 3 different types. You had 0 micromolar, 1 micromolar, and 10 micromolar.
0, 1, and 10 micromolar, yeah.
What did the 10 micromolar?
It was so what I was trying to get at is that if you see the graph in the top right with the toxicity, see how it just shoots down to 50. So that at that concentration of drug, we're having problems, making reasonable conclusions from the data, because at that point we didn't know if what we were seeing, what was from the actual, drug and radiation working together, versus it was just so saturated in drug that it was just killing the cells. So what I mean is, can you see the pointer?
Yeah.
So for this bottom right, so these are the raw values. The graph on the top left, that radiation response curve, that's all normalized to the percent of the non-radiation control. So essentially each of these values, we standardized it to this gray line right here, which got no drug at all. But for this one, at the 10 micromolar, that initial luminescence value, because the idea is that, if you're getting no radiation, they should kind of start off at the same point, but this was like right here. So already without any radiation, that concentration was just plummeting the cell number. So, and that's when I said, if I had more time in the program, we would have left the drug on for maybe, a bit less, or maybe tried a concentration of like 5 micromolar. But, we unfortunately didn't get to that. But, what was good is that with the 1 micromolar concentration, we got the results that, we were looking for. But again, I would love to have had more time to do this. That's the thing that kind of, sucks about these 10-week internships is that, once you get into it, it's like you're almost done. Because I remember hearing a quote that's like, the unit of time in a research career is a decade. So 10 weeks really is, a very short amount of time, kind of get this stuff rolling. But did that answer your question?
Yes.
Or, yeah. Okay.
I'm just guessing right now you're working with cell lines, and it seems like you're working. So I'm guessing the next step will be the method of delivery of the drug. Do you ever?
What do you mean method of delivery?
How would you deliver MPA to?
So that actually brings up a good point that I probably should have brought up. So MPA, the Wall Lab, we're actually during the summer, and is still ongoing, doing clinical trial in adult GBM patients with MPA. So MPA is actually the prodrug of MMF. So MMF cancer, these cancer patients, they take it, it gets metabolized in the liver, and what gets delivered to drugs to the brain is MPA. But again, that brings up a really good point, and that method of delivery is that, I think I forgot to go over, is that the blood-brain barrier makes it extremely difficult to deliver drugs. You know, that's the biggest hampering to delivering chemotherapies. You know, if you're doing it, you know, through the bloodstream to these tumors in the brain. So, you know, we need. I know there's, you know, kind of nanoparticle and nanolipid technologies kind of cross, you know, the blood-brain barrier and hopefully diffuse into the tumor site. But, you know, that's still an ongoing area of research, and the dosages that they get to the tumor, I know, are still not, you know, what is desired. But again, you know, definitely, you know, in patients especially, it's very difficult to deliver those drugs into the brain. I know it's easy to do it in culture when you have no membrane blocking you. But yeah, Anthony.
Is that why you guys are like preferred? Or like it doesn't
It's not like we only prefer to use radiation because it's the only thing that's been shown to work. I know, especially for DIPG, especially for GBM, I know there's a common drug called temozolomide that is used as a chemotherapy, almost always in combination with radiation therapy and surgical resection. What's tough is that DIPG, you can't surgically resect it. Radiation is the only standard of care that's shown efficacy in humans. You know, pretty much every clinical trial for drugs trying against DIPG, they just don't work at all. They don't help in addition. So the effect of radiation therapy plus the chemotherapy is essentially equivalent to the effect of radiation on its own. Yeah. Yeah.
When you were doing this, I'm assuming you did it.
So it was cool. It's like I handled all the in vitro experiments. So like, you know, growing cells in culture, you know, doing the Western blot stuff. The graduate student I was working with, he started a mouse experiment kind of halfway through using this kind of same drug. But he was doing like the in vivo stuff.
My question was like, has it been shown? Because I know you guys said that you.
So we're not doing a clinical trial in DIPG. My PI, he's doing one with our postdoc on GBM using this same drug essentially though. And they're getting pretty good results for it, I think.
Okay.
Yeah.
So 2 questions on unrelated. The first one, the 2 cell cultures that you used were BT2 in your
BT245 and DIPG13.
So they're all brain cells with you.
Yeah, they're all DIPG tumor cells.
Okay.
Yeah.
Perfect.
And then within each cell line, I had a wild type cell line. So wild type BT245, for example, then a BT245 cell line with that H3K27M mutation.
Actually, I have a second question. It's not the second one. So
Yeah.
Question.
So
in your Western blots, that was really nice. You use beta-actin as your housekeeping gene.
beta-actin as the control, for the Western blot,
Why beta-actin and not something else?
because you know every cell expresses it.
Absolutely.
Yeah, you're trying to grill me over here.
You could use GAPDH or
Yeah, I just did a Western. Yeah, I work in a pediatric oncology lab at Dana-Farber right now, and for our Western blots, we were using GAPDH for our control. Yeah.
Okay, and my final one, which is not related to NPA, was the mutation of the histone, the H3K27.
Yeah.
I was wondering if you know if people have tried to silence that mutation by using that like short-haired RNA or something like this. Do you know something?
You mean like in humans?
Yes.
Trying to
Or
target
even
that
like,
mutation?
if they have mice model of the same disease. I was wondering because, especially in the recent years, shRNA has become more and more used just because it's such a new technology. I was wondering if anyone has stumbled on that.
You know, that's something, you know, to be honest, I'm not sure.
Okay.
You know, I'm sure people have tried. I think, you know, these are very difficult to target, you know, because, you know, these epigenetic modifications, histones, you know, they're like, you know, as a slide, you know, suggests, you know, very complex and dynamic. You know, but it does offer the potential for targeted therapies. You know, I would say there's probably more the focus on trying to, I guess, you know, remethylate that area. But, you know, to be honest, I'm not sure.
Yeah.
Yeah. Yes. Mhm.
You talked about only methylation.
Mhm.
I understand that this component get conjugate like they
Right.
but
Yeah.
like a bond completely form, like they get close together,
Mhm.
and in acetylation they free up.
Right.
So if they attack only the methylation thing, they are looking at the say or the gene that is regulated for methylation. Why they need to think of targeting metallics or the methylation enzyme rather than going to the IMPTH?
Oh, you mean targeting that enzymatic complex?
Yeah, that because there will be upregulation of the enzyme more and more and more.
Right.
Stop it. See, if you want to stop the water flow from your tap, right? You should not close the tap. You should close the leak. You won't get water at all. You won't have the pipe
yeah.
issue, leak of the pipe issue, anything else.
I see what you mean. Like the thing is in this disease that like we want that region methylated. So we want that enzyme to methylate that region because that silences those genes. What the mutation does is it prevents the enzyme from attaching those methyl groups. And hence you have the region of the genome that should be silent that's now turned on. Say that again?
So
or
in drug therapy, it's either agnostic or anti-agnostic. So they could have used agnostic therapeutic agents to increase the methylation.
yeah, I mean, yeah, that seems like, definitely a potential area. That's something I haven't looked at that much or didn't come across, when I was reading the literature this summer. Yeah, it makes sense, finding, something to, methylate those regions or, have a, methylating enzyme. It can seem kind of undo that mutation.
My second question would be like myophenolic acid. Do you know why this is normal? What is the application?
What is the what?
Normal application of normal therapeutic use of myophenolic acid.
Mycophenolic acid. It's just an IMPDH inhibitor.
No, it is used in.
Oh, is it?
As immunosuppressant.
Oh, I wasn't aware of that.
So if you give it to an animal study, it will die due to bacterial infection. Getting rid of your IMDPH before getting a cancer cell treatment with radiation. Radiation kills your immunity. If that you're going immunosuppressants, you just die by normal bacterial infection.
Oh, I see what you mean. So with the mouse studies that the graduate student I was working with was doing, they were immunocompromised mice.
Yeah, immunocompromised mice. So like you could say like taking a normal human being and I give myophenolic acid, radiation therapy. Radiation therapy kills the immunity of your body. In that you get immunosuppressants. It becomes totally vulnerable for the patient or the person. Might just die from normal bacterial infection just by like a normal cancer.
Interesting. Yeah, no, that brings up an interesting point. That I didn't hear about that this summer, but I can definitely read about that more.
Last would be that do you think myophenolic acids can pass through blood vessels?
It is blood-brain barrier soluble. Yeah. The trick is it can cross blood-brain barrier, but not in a high enough concentration. So it can, but not all of it can reach the site, or it's not, and that's a common thing across the board for drugs that are blood-brain barrier soluble is yes, they can cross it, but not in the dose that we want that allows it to exert the effect that we want on the tumor. I'm not sure. Yeah, I'm not super aware of the, I guess, medicinal chemistry of the drug. I was only kind of researching from the molecular biological perspective. I do know, like when reading the literature and talking, with other lab members, that it is blood-brain barrier soluble. To which extent, I'm not sure. It's a good question. Yes, Emily.
That when you were doing on for so long period of time, okay?
For 48 hours.
48 hours. Okay. So how was that determination?
I don't know. I think it was just kind of arbitrary. That was, previous studies they would leave the drug on, for, various time points of, 24, 48 hours.
I was wondering if you're trying to saturate all of your cell lines have a lasting effect? And then what would that kind of keep? I was just curious to see if you were like, what kind of route?
I know, like, what it can offer is that, you have to apply the drug before the radiation. Because, the radiation, when it hits the cells, that's immediate. That ionization, occurs right away. But with drugs, especially, ones that's being incorporated into pathways, it takes time to do that. So if you hit them with radiation first and then apply drug after, you couldn't necessarily conclude that the effects you were seeing were in the context of the drug being there. So like, I remember actually asking that question. I was like, why do we actually apply drug first? And my PI explained it. He's like, you need to give the drug time to incorporate itself in there. Then you irradiate, when the drug's already there and kind of, working its magic, then you see how it affects that response. Specifically like the time points, like I know in the lab I'm working with right now, like we apply drugs and like we do time points at 24, 48, 72, 96 hours.
So was there no cell penetration assay then?
What's that?
Cell penetration assay. You a cell a time and you isolate the cell by cell lysis, then you do a centrifugal test and find the amount of drug passed through the cell. Was there any study done with that? Or
We didn't do a cell
penetration assay?
penetration assay. We just apply the drug, for 48 hours and then irradiate after. I know other lab members might have been doing that, but where? Yes, Andy. What do you mean? What type of radiation? I think it was just ionizing radiation. Like we just had the same type of radiation as a patient would receive.
I don't know much about radiation or barium
I know that like I don't know. I think it was like electron beam or something. Like whatever is used against patients. Like we put the cell into this irradiator machine. It would just kind of hit them with radiation, but it was the same type of radiation that patients would receive. But the exact type of radiation, I'm not sure. I don't know if there's like different types of radiation used for use in clinic, but yeah. I know there's, kind of internal radiation that, you take as, a therapy and it, irradiates, from within. And then there's obviously the external beam radiation, which, you kind of fire and it's the cells like that. But. Yeah, not in any meeting I had or shadowing the radiation oncologist I was working for, like when he was going over radiation treatments, there was no mention of the exact type of radiation. It was just, what they were just talking about radiation being applied. The exact type I couldn't say, but I think it was the same. I know the same type of radiation, especially he used because all of his patients had high grade brain tumors. It was the same radiation he was using. But again, like the exact type of like iodine, cobalt, I'm not sure. So that's actually a really good question. And it was interesting. I'll kind of backtrack. So that's what you kind of wear for radiation. It's actually really uncomfortable for patients. It's claustrophobic, and they make this kind of plastic mesh, mask that you kind of have to, sit still in for, half hour. And then there's, this machine that kind of revolves around you and like fires the beams of radiation. It was actually really cool. Like when I was shadowing my PI, he was showing me how he plans his radiation treatments. And it's this really cool kind of computer software. It's almost like a three-dimensional image of like a CT scan or whatever. And, he actually physically, with his mouse, directed where he wanted the beams of radiation to hit, so you would kind of hit it from, if the tumor is like here, you'd hit from the top, from the side, from under. And the idea being you want to get as much you want to hit as much tumorous tissue as possible and while avoiding as much healthy tissue. But that was pretty cool to see. Yeah, thank you. Thanks for coming. Thank you for coming. Thank you. Cool. Thank you. Thank you very much, Dr. Shia. Thank you all for coming, especially those on Zoom. Oh yeah, does anyone, oh, there's a Zoom chat. Maybe there's a question there. More chemistry research, more biological research for you in the future. Aha, that's a really good question. They're both really interesting. You know, so far I've only, the labs I've worked in have only been more kind of molecular biology, but I do find myself really interested in kind of the chemical aspect of kind of drug synthesis and design. I went to a talk this summer at the University of Michigan with a pediatric oncologist from Seattle Children's Hospital, and his lab was working on synthesizing novel, peptides and proteins to be used to target cancer and actually developing drugs. And his background was in pharmacology, and I thought that was a really unique perspective. It's kind of more at the drawing board and more creative aspect. I would like to experience it first, working, doing kind of basic organic chemistry research, in the context of drug design and development. But they're both really cool areas. Cool. Thanks for your question. Cool. Well, should I stop sharing the screen? Okay. Thank you.